Okay, this is Motor Control Lab 1, Part 1, looking at patellar tendon reflex. So we're going to examine that typical reflex when a doctor taps your kneecap and the knee swings out, and see sort of what they're looking at, what they would be looking for, mainly amplitude, or the amount that your knee would kick out. That's the amplitude of the response. And then latency is something we can measure with our signals, but we'll get a little bit more into that in a second. So first we need to understand what's going on from our sort of textbook theory side of things. So in our quadriceps muscle, which is attached to our patellar tendon, we have muscle spindles which detect stretch. So we have regular muscle fibers which are innervated by our alpha motor neuron here, which drive regular skeletal striated muscle contraction. But then you also have muscle spindles, which are wrapped around intrafusal muscle fibers, which can regulate the actual amount that they stretch. So they're actually governed by gamma motor neurons. We won't talk about that right now, but just know that when these muscle fibers, these specialized muscle fibers, are stretched, they're wrapped around by this specialized sensory nerve called a 1A afferent. And that 1A afferent has its cell body up in our dorsal root ganglion here. So it actually is a bipolar neuron, or pseudo-bipolar. So it has a signal traveling back up towards the spinal cord. Typically, you would think if you have descending input from your brain going down to an alpha motor neuron and then driving skeletal muscle in terms of like voluntary drive, this is going back up into the spinal cord. So we have our sensory afferent information going back up into the spinal cord here. This is what we're mainly concerned with, the response to the homonymous muscle. So we have a branch of the 1A afferent coming off and directly exciting. So this is a excitatory synapse here. We have it directly exciting the homonymous alpha motor neuron, driving that same muscle, the quadriceps. So when this muscle gets stretched as a reflexive or protective mechanism, it contracts. And then in order to sort of preserve that muscle and have it not get overstretched or stretched in a way that is unexpected by the body. And then a sort of side branch of this 1A afferent goes to what's called a 1A afferent interneuron. This is actually an inhibitory interneuron. So this is an inhibitory pathway going to our antagonist muscle, actually inhibiting our hamstring muscle. Because if they were both excitatory, you would have steady co-contraction, sort of like joint stabilization, but what the muscle is trying to do is get this protective mechanism and contract the muscle. So when we have a tap, an unexpected stretch, you have a direct excitation of that same alpha motor neuron, or many in this case. This is just a diagram of one, but it would excite, you'd have multiple 1A afferents exciting the majority of the sort of high threshold neurons in the pool, and then activating that muscle. And then you'd also have that inhibitory response of the hamstrings, allowing the knee to actually functionally kick out. I'll talk just really quickly about what our intrafusal muscle fiber looks like, or our muscle spindles. So this is the whole muscle spindle. You can see we have our intrafusal fibers here. They're situated inside this capsule, and the afferent axons actually run into them, wrap around the intrafusal muscle fiber, and then when this is stretched, that's when they depolarize and will send that sensory signal back up towards the spinal cord. You see here we have efferent axons. So these are not alpha motor neurons. These are what I was talking about, are gamma motor neurons. So they regulate the sensitivity of this whole system. So it can actually get more or less sensitive. You don't need to worry about this too much, but that's sort of a nice overview of what happens when these actual muscle fibers are stretched, and they're embedded in the muscle. So when the whole muscle stretches, these stretch as well. Okay, so we're going to get into our lab protocol now. I'll play this video and just sort of talk over it, making sure that we walk us through sort of the pre-lab setup, everything you need, and then I'll do a separate analysis video that will follow this up. Okay. So yeah, the first lab is called Reflexes and Reaction Times. We're in the reflex part, but I'll talk about the reaction times part as well coming up in a second. This is testing that monosynaptic, so one synapse Patellar Tendon Reflex. This is our required equipment. We obviously need our lab chart software, but mainly our computer cart and our power lab. This specialized unit here acquires all our signals, and it has a little amplifier in it as well. We need our goniometer. So what that does is it can measure the flexion of the knee. So this is a digital goniometer. You're probably used to an analog one, but essentially when it's bent, it changes the amount of voltage that comes through. This is a little specialized tendon hammer, different than what the doctor would use. So on the tip, it actually has a sensor, so we know exactly when it strikes the patellar tendon. We need this little adapter to connect it because it has a specialized DIN connector. It doesn't connect via the regular cable. We need this little finger pulse transducer for our experiment number 2. And then this little push button switch, again, for our experiment number 2. That's the reaction time part of the experiment. And then finally, we need some medical tape. I implore you all to read the pre-lab. It has more content, kind of like how I described, in better words, not sort of talking off the cuff like I am. So the more you read the pre-lab, the more you'll be prepped and sort of understand the lab when you're ready to go. It again will talk a little bit about the monosynaptic reflex like I did, but it really, in terms of understanding it and then going through step by step the protocol, the more you read pre-labs before and understand this video before I actually talk you through it, the more you'll sort of understand what's going on. And again, just one last refresher, we're going to be tapping the patellar tendon, but this time our tendon hammer will actually send a signal back, tell us exactly when we tapped it, and then our goniometer, which is going to be attached to the knee on the side here, will tell us exactly how much the leg moves, but we can also see the latency of the signal on when it moves. So we're going to strike, we're going to have that signal go back up through our 1A afferent, activate our homonymous alpha motor neuron, then come back down, activate the muscle, a little mechanical delay, and then extend the knee. So we're going to be able to look at not only the latency of this reflex, but also the amplitude of it. So how many muscle fibers is that activating? And is this 1A afferent pathway more excitable, so it's activating more alpha motor neurons, or less excitable, so activating less alpha motor neurons, and therefore, because it's a 1-to-1 ratio in a motor unit, less muscle fibers. This is what we're going to need. We're going to go in and we're going to grab our equipment. So like I said, we're going to house it in these boxes, have it on the tables ready for you guys. We're going to pull out our tendon hammer first and our goniometer. This is this little reflex kit, and we're going to have some zinc tape here. You can use medical transport tape as well, but these are the 3 things you're going to need. You can notice here that it doesn't have that little B and C adapter on the end, so it doesn't look like this connector that will fit nicely in the power lab, so that's why we need that B and C adapter to make sure. So it translates this connection into one that's more easily readable by the power lab initial analog inputs. You see that we have our power lab case here. We'll label them if there's anything up with them. This one says broken hinge inside, so we know to watch out. But typically they have every piece of equipment we would need. They're labeled power lab 14, so please don't switch items between them because we catalog everything that's inside. So if we open the case up, you'll see that our power lab is missing, and the power cable and USB cable is missing as well. But if we flip open the top, then we can see housed is our little push button transducer. So what that does is you use your thumb to press on the top, and it basically sends a signal just like a regular old sort of light switch type of deal, a little bit more sensitive. And this is our finger pulse transducer. So it's a very similar type of deal, but it can detect amplitude a little bit, and it's a more sensitive membrane. It's not like a very clicky membrane. So going on to our power lab on the cart, we want to reach around back and turn the power on. A little green light will come on the front. We want to make sure that our tendon hammer has that input plugged in, and then we're going to plug our tendon hammer into analog input 1, making sure that it's nice and sturdy in there. They're these little 8-pin DIN connectors, so making sure that you don't bump it too much, you're going to plug in the digital goniometer to channel 2 and attach the goniometer using the Velcro straps on the side of the leg, making sure that the person is sitting sort of regular on the table, not with their feet on the ground and not way too high up. You want the knee to sort of be swinging freely, because when you tap the tendon, you want it to be able to elicit that reflex. Then on the computer, we're going to go into the PC, navigate to our local our C drive, users, scroll all the way down. These are all the people's profiles who have logged in before, but we put all our data settings files in the public section. So we're going to open our knee-jerk settings file. That's what we're doing. It's named appropriately. So it's going to look like this when you pull it up. So on the top, we're going to have our tendon hammer, or our sort of stimulus channel. It will drop a little line exactly at the moment we strike the tendon. And then on the bottom, we're going to have the goniometer, or the response. This is what it's going to look like. Takes a little bit of practice to exactly hit that tendon accurately and elicit a big response. Let some time in between, and you won't always get good ones. You can go back and delete them if you don't get a great one. And this is where commenting out your data really helps, and making sure that you palpate the tendon and you can stay as consistent as possible. We don't have it pictured here, but I would maybe even put a little mark on the knee and stand over the person like a little bit closer, and then you'll just sort of get the hang of this as you go through. This is what it looks like as you're actually collecting data. So once you've practiced that enough, you're going to hit start. And then as you tap with the tendon hammer, you can see the strike from the tendon hammer here will actually automatically zero that. So it's collecting data all the time, but it drops the zero marker at the instant where the tendon hammer strikes. So once we've collected our 5 trials here, we can then add a comment by right-clicking and then typing regular, because we were just striking it. The person wasn't doing anything, they were just relaxing. Following that up, we're going to get the person to perform what's called the Transducer maneuver, where they link their hands and pull apart as hard as they can. Then what we're going to do is we're going to collect 5 more. So again, using our tendon hammer, making sure that we tap the knee, give some time in between. Kennedy's looking pretty concentrated there. So then making sure, again, once you've sort of practiced that once, you're going to hit start. You're going to do it 5 more times, this time with the person performing that Transducer maneuver the entire time. And once we collect all those, we'll go into make sure that we comment out which ones are Transducer, because we're going to need to know that for our analysis. So again, we count out our 5, making sure. Add our comment by right-clicking, add comment, type in Transducer maneuver, whatever you want to type in. And then coming over to the left side, I believe you can go into the top left right here and click that little button and click auto scale. Or what you can do is you can double click the axes here, and what that will do is make this a lot bigger. Okay, so instead of doing the auto scale and sort of orientation in a previously recorded video, I'm just going to do this live and make sure I can walk you guys through it. So I'll do a quick orientation to LabChart software here. If you're online, you're going to be using LabChart Reader, which doesn't allow you to collect data, and it might not have the exact same options, but the sort of general layout and idea is the same. So we have our signals here that we acquired in our channels. So this is where you label your channels. There's 2 channels present here. And if we go into setup, channel settings, you can see which inputs are corresponding to what channel. So here it says stimulus, that is actually our tendon hammer, and 2 is angle, that's our goniometer, but it's a sort of more relevant explanation of it there. Here I have my start button grayed out. That's because I don't have a Power Lab plugged in right now. For LabChart Reader, that won't even be an option, because Reader is just designed to analyze data after the fact, which is perfect because we're online and you guys don't have a full LabChart 8 license. So the first thing I sort of mentioned was auto scaling. I think that's important, being able to scale and see your data appropriately. So the first thing is auto scaling the y-axis is probably one of the first things I do. You can do that by clicking this little arrow up here and clicking auto scale. That will automatically take sort of your largest amplitude, your smallest amplitude, give you a little buffer on either side, and make those and optimize for that Y range. You can also grab and drag anywhere on these axes. If you grab high, if it will drag smaller. If you grab low, it will do the same thing from the bottom side. And if you wait for that sort of in-between arrow facing both ways, you can adjust the baseline and slide it up or down to get a better view if you want to look at very specific data. But if I want to go back to auto scaling it, the second type of way I can auto scale it is to just double click the y-axis. And Oh, it's apparently zooming out right now. Okay, there we go. So I guess it has sort of 3 functions where it'll zoom out and then it'll auto scale for you. So that's auto scaling in your Y, and then you can see that this data set is a little bit different than the one I had in the previous video, but we do have our data commented out. So we have end of our 5 trials commented out there. And the other thing we want to do is maybe auto scale on the x-axis here if we think that these are a little too bunched together. So here we have our ratios. So if I go to a 50-to-1 ratio in terms of time spread out on the screen, you can see it gets a lot smaller. I think we were at 5-to-1 before. If we go to a 2-to-1, what I can do is really zoom in, and we can see here the electrical artifact that was left by the tendon hammer. So right at this point, if you can see my cursor here, is when the tendon hammer strikes. And you'll notice that you have this little X mark. As I drag my cursor left to right, my cursor doesn't have to be in the waveform, but this X mark will trace along the waveform wherever your signal is. So you're going to have a certain signal amplitude. That's sort of the inherent reason we're collecting this, is to get changes in these signals. And you'll see that this waveform cursor will slide along it the entire way. Now, what this waveform cursor does is it at any point you can then come up to the top here, and you can see it says no sampling. And it's going to be hard for me to do this with only having one cursor, but you can see where it says no sampling here. If I put my cursor back on the screen, you can see that the top channel, it's reading negative 0.001 volts. Again, this is in volts because really we just care about the latency at the time the tendon hammer strikes. And then the bottom one is in degrees. So you can see that if I put my cursor right here, it's 32.84 degrees. Now that's not necessarily normalized to the person's knee. We think it would probably be pretty accurate, but what we're mainly concerned about is the change in knee angle, not the actual knee angle itself. So we're going to be referencing these back and forth to each other. So really what we're going to do here is in terms of latency for this experiment. So the first thing we'll ask you to do is find latency. You're going to go through your trials, taking your waveform cursor, going right from the time when the tendon hammer strikes. So you can see up at the top here, it will give you time. So if we go right from the time when the tendon hammer strikes, and it will, at this one actually, the peak will right there, right when it starts, right when it goes above baseline, this automatically levels it to 0. That's sort of like the function of the tendon hammer. So you actually don't have to go in and look at the exact time when it strikes, then look at the time when you get the reflex response and subtract it. This is nice because it's sort of automatically zeroed for you. In other experiments, you will have to go in, get one time, then reference it to another time and create a function later. We don't have to do that here. So what we're going to do is just go to the time when we see the response from the quadriceps muscle extending the knee. So I think we can see a response right about here. I'm going to then look at the top. It says 0.054 seconds. And what I'm going to do is I'm going to go into an Excel worksheet, which I've created that looks pretty much like what is in your workbook there. And that was for the very last trial of them all. But I'll write 0.054 seconds. And we can even make sure that we want to know that this is in seconds so we can remind ourselves of that. You'd go through, you'd write all these in. So for this one, you'd probably have right about 0.5 for our first one, 0.05, sorry. For our second one, when do we start? 0.062. And then see how it's sort of almost too zoomed in for me to work here. If I have them side by side on the screen, that's when you would use this X scaling function and zoom out a little bit just to give yourself a little bit more room. It doesn't allow you for that accuracy, especially when you're picking out very specific points. So I've done the first 2 trials, and I'm doing the third here, 0.055. Do the fourth. 0.065, and we'll do the final one here of our regular. Call that 0.055 again. So I think they actually, oh, see, they had a bad trial in there, and then they just commented out that it was the end of it, so they left it as the 5. So then what we can do is we can calculate our mean, knowing that in Excel, if you press equals and then start typing average, we can get a total average, and this is more sort of stats functions, but we want to get just a regular average. In our brackets, we can then either write our cells or drag across all the cells we want to average. You can either close the brackets manually, or if you're done with a function, this is a pretty simple one, you can just hit enter. We then have our mean calculated for those 5 cells. Then if we want to calculate our standard deviation to give us a little bit more information about this, we can type in stdev. Doing standard deviation of the population is a slightly different calculation, which you'll know from stats. So we want to do standard deviation of the sample. Double click on that to get it to auto-populate, drag and drop, and then we have our mean and our standard deviation. So that's what you'll do. You'll go through and do that for the Gendrastic maneuver as well. We have our normal up here and Gendrastic down here. Then the next thing we want to do is look at the change in angle. So this is where we're going to use this little marker here, because if I just come up here, I can see that my angle is at 59.3, again referencing the number that's right here because we're on channel 2 now. So I can see my baseline here is about 55-ish, and then it takes a little peak there. Let's say my baseline is 55.8, and then I know that the peak, or the total amplitude of the response, the angle decreases to 48.4. So if we can flip this goniometer around, we would have an increasing angle. They had it flipped a certain way, so it's a decreasing angle. We really just care about the total amplitude, the absolute amplitude of the response. So instead of writing down 55.8 and 48.5, what we can do is use this marker down here, this M. If we drag that M up, it gives us a point of reference, and now you can see over here, when I have my waveform cursor, it is only referencing a change in signal to where that M is located. So now if I sort of seek out the bottom of that signal, I'm again looking for the peak, so I'm looking for the most negative or biggest change here. It looks like we got negative 7.34. So that's going to be our first one, negative 7.34, and then we're going to do the same thing. You can get rid of this M by double clicking it and then bringing it back up, or you can drag it each time just between where you want to drag it. So the next one we're going to hunt out, so that was a change in 4.04 was the biggest one I saw, negative 4.04. Bring our M over, negative 5.33. Bring it over one more time. Nope, 2 more times. Second last one, negative 4.95. And one more time here, because we know that the 6 isn't a good contraction, 5.55. 5.55. Ooh, negative 5.55. Okay, so now in your chart, if you have your mean here, you should probably know this, but if you copy and paste this, this cell, you're going to get a different mean here. And if we click on this cell to see what it's referencing D3 to D7. But if we click on this one, it's E3 to E7. Because these haven't been cell locked, because I slid this equation over one cell, it's going to slide over everything it's marking as well. If you want to see what a cell is measuring in an equation, you can either double click on it. It will light up in a different color depending on how many embedded functions you have, the cells that are highlighted. Or if you have you can always press escape to get out of that. If you really want to take a look at what something is doing on your keyboard, you can do control, and I don't know what this is called, but the squiggly mark under escape will then type out the cells. It won't give you the function. It will type out what the function is and not give you the result of what that function is. And then if you want to get out of, so you can really easily see, oh, this cell is referencing the average of these, and then it's going to output that, and then this is going to be the standard deviation of those, but then this is going to be average of E3 to E7. If you want to get out of this, you can just control squiggly mark under the escape button again, and it'll take you back and actually give you your numerical results again. So we're going to copy and paste our standard deviation under our angle. So now we have our results here, which I will bold, and that is what you are submitting for the first part of lab 1, and that should give you, without giving anything away, some inference about what the difference is between a normal latency and normal max amplitude of a knee tendon tap reflex and a latency and max amplitude when performing the Gindrasic maneuver. Again, I know what's sort of going on behind the scenes with the Gindrasic maneuver, but without giving anything away, I'll let you go ahead and research that and see why there's a difference between the two. And then this is where we sort of get into melding you guys looking at the data, interpreting it, and then linking that back to the theory, and then making sense of it that way. Thanks for watching, and have a good one.